Maternal effects are an important exception to the traditional view that an offspring's characteristics are determined solely by the genes it inherits from its parents. In a maternal effect, the mother's genotype, physiology, and environment influences the offspring's early phenotype independently of the offspring's own genotype. Instead, the offspring initially depends on factors provided by the mother before its own genome becomes active. Maternal effects arise in two ways. The first involves maternal effect genes. During oogenesis, maternal effect genes are expressed in the mother's ovaries, producing messenger RNAs, proteins, transcription factors, and other regulatory molecules that are deposited into the egg. After fertilization, these maternal gene products direct early embryonic development until the embryo undergoes the maternal to zygotic transition, MZT, and begins using its own genome. Because these molecules are produced according to the mother's genotype rather than the embryo's genotype, the offspring's early phenotype reflects the mother's genotype. A classic example of a maternal effect gene occurs in the freshwater snail Limnaea. Shell coiling is determined by the mother's genotype rather than the offspring's genotype. Mothers carrying the dominant allele produce offspring with dextral or right-handed shell coiling, whereas mothers homozygous for the recessive allele produce offspring with sinistral or left-handed shell coiling. As a result, an offspring's own genotype does not determine its shell coiling, but instead affects the shell coiling of its future offspring. This one-generation delay is a hallmark of maternal effect gene inheritance. The second type of maternal effect results from the mother's physiology or environment. During egg formation, factors such as the mother's nutritional status, hormonal balance, age, and overall health influence the composition of the egg. For example, a well-nourished mother can produce eggs with large nutrient reserves, giving embryos more resources for early growth and development. These effects arise because the mother's physiological condition influences the composition of the developing egg rather than through differences in the genes inherited by the offspring. Unlike maternal effect genes, these maternal effects can vary from one generation to the next because they depend on the mother's physiological condition and environment rather than her genotype. Maternal effects are often confused with other forms of non-Mendelian inheritance, particularly cytoplasmic inheritance and genomic imprinting. But they are distinct. In cytoplasmic inheritance, traits are determined by genes located in extranuclear DNA, most commonly in mitochondria or chloroplasts, which are transmitted through the egg cytoplasm and inherited directly by the offspring. In genomic imprinting, certain genes are expressed differently depending on whether they are inherited from the mother or the father because one parental copy has been epigenetically marked and silenced. The offspring's phenotype is therefore determined by the expression of its own inherited genes. In contrast, maternal effects result from molecules supplied by the mother or from her physiological condition during egg formation, influencing early development before the offspring's own genome assumes control. Maternal effects are temporary because the influence of maternally supplied molecules gradually declines as embryonic development proceeds. Once the embryo's genome becomes active, it begins producing its own RNAs and proteins, and control of development shifts from maternal factors to the offspring's own genes. However, maternal effects can still have lasting consequences because early developmental events often influence later growth, physiology, and survival. Maternal effects demonstrate that inheritance is influenced by more than the DNA sequence inherited from both parents. The mother's genotype can shape early development through maternal genes, while her physiology and environment influence the conditions in which the embryo begins development. Although maternal effects are the best known example of parental effects, fathers can also influence early development because sperm deliver not only a haploid genome but also RNAs, proteins, and other molecules that can influence early embryonic development, while seminal fluid can alter the mother's reproductive physiology. However, maternal effects are generally much more prominent because the egg provides most of the cytoplasm and developmental resources required for early embryonic development. Unlike cytoplasmic inheritance, maternal effects do not involve the inheritance of genes carried in extranuclear DNA, and unlike genomic imprinting, they do not involve parent-specific expression of the offspring's own genes. Understanding maternal effects helps explain why offspring with the same genotype can differ in their early development and highlights the important role of parental influence before the offspring's own genome takes control. Now test your understanding by trying the questions on this topic. If you enjoyed this video, don't forget to like, subscribe, and share. Thanks for watching.